Hi everyone, I'm Olivia Hatcher, and as the slide says, I did absorption and fluorescence spectroscopy at UCSD in a biochemistry lab. Okay, so this is over here on the left is the UV absorption machine. So basically, light is shot through a slit, and then it measures the difference between the light that is shot out of the slit and measured at the end, and from that you can determine the concentration. So I was using that for all the different molecules that I worked with, which included FITC, which is a fluorescent dye, L-trip, NADA, and indole, which we had like an ongoing joke in my lab that it smelled like feces. Personally, I didn't think it smelled like feces, but there were some people in my lab who did. And then here is just a sample of what all the different dilutions of NADA concentrations and all the different molecules look like. And a big part of my internship was just finding the linearity of the different concentrations of the molecules that I was working with. So these are some of the graphs that I made. Over here, that is an absorption graph. As you can see, kind of at the top there, it goes all crazy, which is not what it's supposed to do. So I actually learned something new and taught the lab that above absorbance of 3, you really can't measure anything accurately, at least for NADA. Here is what the orange line here is what NADA should have looked like in its concentration linearity, and the blue line is what it actually looked like. So we determined that the instrument was not perfectly linear as it should be. I also did a lot of fluorescence, which measures the amount of photons that are emitted once you hit the protein sample or molecule sample with light. So as you can see, there's a big peak at around, I can't read the number, but I believe it's 280. And then over here is just another graph of some of the raw fluorescence data. And as you can see, it peaks again at 280. Some more graphs. A lot of my internship was graphs. This was the fluorescence machine. So as you can see, there's a dip here, a little divot, which was not, again, supposed to happen, so we learned something new. And I measured all of these to get the correct fluorescence of them. And this is FITC, which is a really fluorescent dye. I think I have pictures of it on the next slide. And basically, I used it to measure the linearity of the machine. And so the yellow line here is what the orange line should have looked like, which it didn't. And then same with the gray and the blue. So as you can see, it wasn't linear. Okay, so this is the FITC. This is it in just regular light in a Falcon tube. And then this is it being hit with 280 nanometer light, which is with this really beautiful green. And then this is just with another, so this is, the FITC turned green when it was hit with blue light, and this is just showing that it was hit with blue light, and as you can see, it's a little green around the edges. And then my final project was working with cytochrome C, which is a protein that is involved in your mitochondria and making energy in your cells, so very important. This is just a little picture of it, and it's this beautiful red color because it actually has the same heme group, which is the same iron group as your blood, so that's why it's red. So this is my stock concentration, and then this is it diluted. So this is how I was measuring the cytochrome C. I used a refractometer, and so this is what I was looking into, and that's what it looked like on the outside. I spent like 2 days straight looking into this thing, and it really hurt my eyes, but it was so worth it because of just figuring out how the refractometer worked and just learning more about the protein, which was really cool. And then this was to use to determine the concentration. And so that was really cool in a different method than using the UV absorption machine. So this is what I did with my protein. I denatured the protein, and you can't see over there, but the y-axis is the percent unfolded, or the fraction unfolded of the protein. So as the concentration of urea, which is the denaturant I used, got higher, more denatured, until it eventually flattened out because everything was denatured. And from that, I was able to determine the Gibbs free energy, which is basically its tendency to unfold or fold within urea, which was really cool. And another fun fact about urea is that we don't actually know how it works, how it denatures proteins. There are 2 options that scientists think. They think it either affects the solution which the protein is dissolved in or affects the protein themselves, but we don't actually know how it works. And then outside of my internship, I stayed with my cousins for the first 3 weeks, so I saw the famous Midway ship and this famous rendition of a picture taken after VE Day, or VJ Day, victory over Japan after World War II. These are my cousins, and we went out on the longest pier. This is me at the beginning of the pier, and it goes all the way out there, one of the longest piers in San Diego, which was really cool. And it was at sunset, too, so really beautiful. We also had the ability to go to Coronado, which is where they filmed Top Gun, so that was really cool. So we took the ferry there. This is me with my host mom and my 2 cousins, and then they also took me to the world-famous San Diego Zoo, and the koalas were my favorite. The picture doesn't show it, but they were really active that day, which was super cool. This is Logan. He was my favorite koala. And then for the last 3 weeks, I spent time with 2 other interns, Canyon and Karis, who I think left. And this is the view from the Birch Aquarium, which is in La Jolla, which is just gorgeous. So if you ever go to San Diego, go visit the Birch Aquarium. And then we also went to the boardwalk on, I think, Mission Beach. And so this was one of the rides, and it was really fun. And it was like a big giant swing that turned. It was really fun. And then I would like to acknowledge Pinhead, my professor, Judy Kim, and the entire Kim Lab group. And my advice to future Pinterns is be confident. They know that you don't know anything, so it's kind of a spin on fake it till you make it. But just be confident in your abilities. You wouldn't be here if you couldn't do this. Explore in your internship. I had this, some of the grad students at my lab were working on this big laser in the lab, which I didn't get to use, but was really cool, and they explained it to me, which I wouldn't have known if I didn't ask about it. And then the other thing is work hard. You want to impress your mentors and impress Sarah, and so just work really hard. So I think that's it.